So this is going to be the first lesson of our very last unit. It's going to be a very short unit on population dynamics. And this first lesson is on the characteristics of populations. So population size, density, and distribution. In order to study the populations of a species, we look at a few characteristics, like the population size, the density, and how they're dispersed. Population size is usually just an estimation of the total number of organisms within a habitat. It's very difficult to count every individual within a habitat, so we have a few methods for giving a good estimate. Population density is the number of individuals of one species per unit area. An example that I've got here is 300 deer per hectare, but we could be looking, as big as hectares, acres, square kilometers, square miles. Or we could be looking as small as per meter squared if we're looking at something small like an insect, or we could even be looking per milliliter if we're looking at, say, protists in a certain body of water. So two methods for estimating population size and density include quadrats and the mark recapture method. Quadrats are most useful for stationary species or small sample areas. The population of a species is physically counted in each section of the quadrat. Then the average of these counts and the area of the quadrat is used to make an estimation of the total population. We'll see an example in a minute, but I just want to show you there's a few different ways that quadrats can be applied. In cell biology, we use a modified version of a quadrat in order to count, say, in this particular slide, we're counting white blood cells. Quadrats can be used on aquatic species. Quadrats can also be used on a larger scale, like here we've got a mapped out grid over an entire large area of land, which could be surveyed by, from the air. So here's an example of working with quadrats. Now, if you didn't print out the handout for this lesson and you're not following along there, you're going to want to pause the video and write down this question before we get to the solution so that you can follow along. Okay, but here's the example. If students want to estimate the population size and density of ragweed plants in a field that measures 100 meters by 100 meters, the students randomly placed 3 quadrats. Each quadrat is 2 meters by 2 meters. And in each of the quadrats, they found 18, 11, and 24 ragweed plants, respectively. So the first question that we can answer is, what is the average population density of the ragweed plants? And then for B, what do we estimate the population size of ragweed plants to be in the entire field of 100 meters by 100 meters? So we're going to try to figure out how many ragweed plants there are roughly in an entire field based just on these 3 squares that we're placing randomly within that field. Here's how the solution works. The formula we need is the average density equals the total number over the total area. Now this is really similar to another density formula that you guys worked with way back in grade 8, which is density equals mass over volume. So in this case, we have density equals number over area, or N over A. So if we input the numbers that we have 18 plus 11 plus 24. Those were the number of ragweed plants that we counted in each of our 3 quadrats. Now we have to divide that by the total area. We had 3 quadrats. Each of them were 2 meters by 2 meters. So we have 4 meters squared plus 4 meters squared plus 4 meters squared. So when I add all those numbers up and do my division, for B, where we're trying to then take that average and apply it to the entire field, we have our formula, density equals number over area, but now we're going to rearrange it. We're looking for the total number, which is now equal to density times area. So now the density that I calculated was 4.42 ragweed per meter squared. The field was 100 meters by 100 meters, so the area now is 10,000 meters squared. If I take the average that I calculated and I multiply it by the entire area, I can estimate that there's around 44,200 ragweed plants in that field. And that's not a bad estimate based on the size of the field and based on the sample size that we took. Now, how could we improve that number, right? Because as scientists, we want to make sure that that population density and that estimated population size is as close to the real number as possible. Well, the accuracy of this number would improve with more quadrats or larger quadrats. Also, we have to make sure that we're placing those quadrats randomly spread out throughout the field. So the idea here is the larger the sample size. With more quadrats or larger quadrats, we're sampling a larger area. So we have a bigger sample size, and a larger sample size is going to improve the accuracy of our numbers. Let's look at mark recapture sampling. Now, quadrats are very good for populations that are going to stay relatively still for you to sit there and count them. But in dynamic populations, which are moving around, it often becomes impossible to physically count the individual in a given area. As well, you might run into problems where you're counting the same individuals twice. So mark recapture sampling is a method of estimating dynamic populations. Let's look at an example. So mark recapture sampling. A particular lake contains 3,000 bass, and 300 are collected, tagged, and released to mix in with the general population. That means about 10% of the fish within the lake were tagged. After a few days, we collect 300 bass once again. We would expect that about 30 of those bass would be tagged if everything was collected randomly, because the 30 bass represents 10% of the second sample that we took. So that method could be used in order to determine the total population size, or that 3,000 bass that exist within the lake. So here's, on the next slide, we'll look at the formula. So here's the formula that we use when we're doing mark recapture sampling. So it's basically an equivalent ratio. The total number of marked individuals, so that's capital M, divided by the total population within the area, that's capital N, is equal to the number of recaptures, so that's marked individuals that you recapture when you come back later to capture a second sample, divided by the size of the second sample, or little n. Let's look at an example. So let's look at mark recapture sampling. In a 250 meter by 300 meter bush, there are an unknown number of toads. On Friday, 26 toads were caught, tagged, and released. On Sunday, 21 toads were caught, and 3 of those were found to have tags. So those 3 were ones that you had already captured again on Friday. Those are the unlucky ones who got caught twice. So A, roughly how many toads are there in the entire bush? And B, what is the population density of the toads? Here's the solution. What are we given? Well, the total number of toads that we marked, that's capital M, that was 26. So on Friday when we first went in there, we marked 26 toads and we let them back out into the wild. How many toads are there in the entire area? We don't know. That's what we're trying to figure out. Now, when we let them all spread out randomly and mingle with the population, we came back a couple of days later and we caught 21 toads. Out of those, how many were already tagged? 3. So the little m, the lowercase m here, is the number of recaptures, the ones that were captured twice. Now, what was our sample size on the second day? 21. That's our little n. So the calculation is capital M over capital N equals lowercase m over lowercase n, or 26 over the total number in the population, so that's capital N, equals 3 over 21. Well, now we're just going to rearrange. We cross multiply, and we divide, and we get our capital N value, about 182 toads. So we would estimate that there's approximately 182 toads in this area. For B, the population density, we go back to the equation density equals the number divided by the area. We have 182 toads, we assume, over the total area. Now the field was 250 meters by 300 meters. So it's 182 toads over a total area of 75,000 meters squared. That adds up to about 0.0024 toads per meter squared. At that point, it might be better to use a larger area rather than meters squared in order to convey the number of toads. So we might go to something like how many toads are there per kilometer squared, or so on, or per acre. Okay, now how could we improve the accuracy of these numbers? Again, we look at sample size. So the accuracy of this number would improve with larger sample numbers. More toads could be marked initially, and or more toads could be captured in the second sample. The more that we are able to capture, the more accurate our population estimate becomes. If you end up in field biology, you may end up doing some of this mark recapture sampling in university or some quadrat work in university. And there's math that can be done beyond this, where we look at what percentage of the population were we able to survey, and is that an acceptable number, or do we believe those numbers are accurate enough to be published? Okay. So here's a picture of some of these markings that we talk about. So how do you mark a species? How do you mark an individual? Well, here's an example of some marked mollusks. And here we have a whale shark being tagged. And here again, another example. Okay, so what are some of the ethics that are involved when we mark individuals within a population? Because again, as biologists, we want to make sure that we're having a minimal impact on the population. So what are some ethical concerns with this type of research? And how can these negative impacts be minimized? In a class, we'd be talking about that, we'd make this kind of an open discussion. So think for a moment about your own thoughts on what are some of the things that you'd have to consider. For myself, I would say you have to make sure that when you are doing these sorts of studies, you aren't causing the animal undue stress. You aren't harming the animal in your markings. So a lot of the tags and markings that we put on these animals are meant to biodegrade after a certain amount of time. After the study is done, the markings are meant to fall away or disintegrate, so that the ones that we don't recapture, they're not stuck with that tag for life. As well, you have to consider things like, the seahorse in this picture is clearly camouflaged for its environment. Does that bright orange or red tag that's hanging around its neck, does that disrupt its camouflage and make it more of a target for predators in its area? So these are some of the considerations that you have to take into account when doing these sorts of studies. And generally, before a study gets approved, it has to go through ethics committees that are going to look at these sorts of concerns. Now, beyond just population size and population density, we can look at population dispersion. So how are those populations actually spread out? Populations can have clumped dispersions, where organisms are densely grouped in specific areas within the habitat. So here's an example. Clumped dispersions could be the result of a clumped dispersion of resources, such as food or suitable habitat. Could also result from social behavior, such as traveling in herds for safety or hunting in packs. Now I'm going to use penguins as an example here, because at different, the emperor penguin in Antarctica, at different times throughout the season, their population exhibits all of the different types of population dispersions. For example, during the coldest months in Antarctica, the emperor penguins huddle together in one massive clump in order to kind of fend off the cold. So that would be a clumped dispersion. Now, uniform dispersion is where individuals are evenly distributed throughout the habitat. Often, uniform dispersion results when individuals are in competition with each other for food, nesting, or breeding. So natural populations that are in competition with members from its own species will tend to uniformly disperse. those are the species that we say set up territories and defend those territories. Also, humans, when we grow crops, we're creating these monocultures of uniformly dispersed crops. During mating season, emperor penguins go through uniform dispersion when they spread out and give themselves a little bit more space and pair up. Now, random dispersion. The dispersions of individuals within the habitat demonstrates no pattern. Individuals exhibit random dispersion when they are minimally influenced by interactions with other individuals. So, for example, dandelions or sloths are a couple examples of randomly dispersed individuals. They don't compete with other members of their own species for resources, and they don't really impact on one another. So they spread themselves out into a population in any particular distribution. And back to emperor penguins. When emperor penguins are feeding, they are randomly dispersed throughout their environment at that time. One of the very best ways to get an idea of a population size or density without impacting that species in a negative way is by doing indirect sampling techniques. So indirect sampling techniques means looking at the evidence these species leave behind. Like we've got their eggs or their nests. We can see tracks from herds that have moved through an area, and we can determine or give a good estimate of a population size and its movements and its dispersion without actually having to impact on that species at all.